Yes, I'm Jerry Lanfear. I'm the ELIXIR CTO, and I also currently the lead for the ELIXIR Biodiversity Focus Group. This is a group that formed about 9 months ago, and it's tasked with building a strategy for ELIXIR in the field of biodiversity, particularly with respect to molecular data. As part of that, I've been reaching out externally, to understand what current activity is going on elsewhere and maybe who we should be looking at or partnering with. In that context, I met Pier Luigi a few months ago. In fact, we worked together at the Biodiversity Next conference in October, and I realized that he's engaged at a global level establishing standards for molecular data and biodiversity, and that's what prompted this invite to him to share what he's doing with ELIXIR, perhaps as a first step to further partnership in the future. So with that, I'll hand over to Pier Luigi.
All right, Jerry, thank you for the introduction. Am I audible? Everything's clear?
Yes, we can hear you.
Okay, great. So thanks. We'll kick it off then. Welcome, everybody. This presentation is indeed a kind of whirlwind tour of some global activity that's been going on. I've somehow been at the hub of this for a while, basically connecting a whole set of very brilliant initiatives together, and also learning about some operational processes around the world, which really are calling for omics technologies, eDNA technologies to interface with them and come up to an operational grade, transition out of academia, out of a purely innovation space, and sync up a little bit better with operations so that it can be furthered on to other stakeholders. So I'm going to talk about a few things that are really more on my radar. I have a marine bias, working for the Alfred Wegener Institute, also a European bias, but of course this is a global activity and there are many more initiatives out there. If you'd like to know anything about other regions, I'm more than happy to provide you with connections or links as appropriate. But let's jump in. Let's see if I can jump in. All right, there we go. So there's a lot of ways to introduce how we got here, but a large part of it was in my early days as an academic PhD student. I got involved with the Genomic Standards Consortium because a lot of my work was trying to wrangle genomic information in a way to answer very simple questions, but we couldn't because the metadata was all over the place. It would take hours and hours just to find an object or a data object. So I started to think more in the lines of informatics and information theory to how to bring that together through bodies like the Genomic Standards Consortium, either using checklists like MIX to get the environmental and processing metadata right, formats like the biome to package that information to help share it around, and also then broader scale initiatives like Neil Davies running the Genomic Observatories Network, just trying to federate things and on an organizational level, get people synchronized. So that's, I think, the consciousness, like the core consciousness that will carry us through this talk and also through the initiatives. Right now in ABI, we operate a long-term ecological research station up in the Arctic, in the Fram Strait between Greenland and Svalbard, and it's quite an operation. It's quite a multidisciplinary observation framework, and part of that is an omics observation. So an omics observatory, where we gather samples from sediment traps, from sediments themselves, from the water, and we sequence them for eukaryotes, prokaryotes, and other stuff that's in there. Now, that's all good and well. There are many other groups around the world that do this kind of monitoring. But one of the questions that came up is like, how do we interlink? How do we link with each other and start to share data more effectively? And we got involved with a project, a European project called Atlantos. It was a Horizon 2020 project that was trying to pull together all of these disparate observation observatories or long-term stations, which are doing great work, but sometimes are not well integrated. And that integration is necessary if we want to carry them up to the next level, to plug into, for example, the Global Ocean Observing System, the GOOS, or GEOS, or any other initiative. And in fact, our work package leader, when microbial and omic observation was identified as an emerging network that Atlantos wanted to cultivate, I was asked to help that along. And the work package leader was very clear about this. It's like, we need to think more like oceanographers in the way that we approach biology. We need more physical samples exchanged. We need quality controls. And yes, of course, there's a great deal of innovation out there, but there needs to be some sort of stable point so that we can develop things on top of it. Technologists, others can build around that, and also policy developers can react to that in a timescale that's feasible. Because we face a very, very dynamic discipline, and that's both a blessing and a curse. So out of that, while working on this, we published a current opinion paper about microbes, and of course we recognize that omics is much bigger than microbes. But being our focus at AVI, we talked about this. A lot of the material there is applicable to other organisms too. But that was a place where we put together our thoughts and said, okay, what will it take to actually take microbiome research or eDNA research to the next level and consolidate the community? In brief, we saw lots of opportunities. It's an incredible tool. It's an incredible tool to look at life at a very high resolution, but it requires calibration. It requires calibration. It needs more synchronicity between the innovation space, the scientific space, and the operational space where we can start delivering stable data products. We need improved links to observing systems and a way to cope with our rapid technology change. Sequencing technology develops so fast, which is exciting, but at the same time, how do we provide a stable baseline? How do we provide a stable data commons here? So those are some of the things identified in that paper, but that sort of made us think, okay, we really need to sort of pull this together, because if we ever want to see omic observation becoming mainstream, it's not going to be one project. It's not going to be the next huge project that's funded by one nation. It really needs to be a sort of collective framework where those projects can power ahead, do their thing, but feed into something stable underneath it, feed in and build a kind of foundation that we can all use. Right, so that was one of the maps we had where we tried to do a survey and find out who's actually operating in a long-term context with omic observation for microbes. And this is just microbes. And that was quite difficult. It was surprisingly difficult to find out who's doing that. This took a PhD student, Eddie Fadiv, quite a long time to even troll through papers, web resources, etc., contacts, countless emails. And that shouldn't be the case, because what we want to do is move that into a space like Argo, where we can trace, all right, who's sampling when and where, who's, in this case, which float belongs to which nation. And this is run by JCOMOPS, and in a sense, that's where we want to move towards. So we have the technologies to do that. They've been around. It's more like a cultural change that needs to happen, and it is happening. We do see the willingness, and we need to encourage that a little bit more. So that's where we want to get. So, all right, how do we do this? Within Atlantos, we had that opportunity. They said, okay, here's some funding. Let's see if we can pull together some people to help this along and turn this activity into something more collective. So we held a meeting in 2018 in Bremen. We had pulled together quite a few very interesting people from different disciplines, not just academics. So we had people from MBON, the Marine Biological Observatory Network, Smithsonian, private sector individuals were there. Agriculture and Agri-Foods Canada co-sponsored that workshop because they're doing a lot of omics monitoring in soil systems and river systems. It's not just marine, that's just my bias. OBIS, so UNESCO OBIS were there, many others. The Earth Microbiome representatives, Mbari, NEOS, Silva, so some resources that are actually computational, came together. And that was really, really important to have this diversity of perspectives around the table, because this prevented it from becoming overly focused on, let's say, creating another paper. I mean, there are a lot of papers about this, and it's great, but it's more, we had stakeholders at that table who wanted to know, well, what are we going to do about it? What are we actually going to build at the end of the day? And that really helped us focus and also focus our follow-up steps. Then we formed Glomicon as a vehicle to do this. It's the Global Omics Observatory Network. It's grassroots. It was supported a little bit during Atlantos, and now it's more supported by the participation of its members, and it's a kind of collective vision we have. We have, of course, integrated that vision into follow-up grants, some of which have come through, and we'll be continuing it into the future. But I think what this symbolized was that there's a general willingness, and we just needed let's say, an alignment of vision and some activities to bring people closer together. So Glomicon was developed. It brought together some of the usual suspects in the marine space, and the Genomic Standards Consortium too, but also others. So NOAA's involved. Kelly Goodwin was represented, gave a remote talk there about their initiatives and their work with eDNA and monitoring coastlines in particular. The Earth Microbiome Project, connecting that to a legacy of where do we go from something like that very ambitious and very successful project. And other standards agencies, so not just genomics, but biodiversity information standards agencies also came along. And it's been growing ever since. More interest has been coming in from multiple partners, because again, everyone wants to tap into this, but regardless of how successful any given project is, the outcomes tend to evaporate unless it's feeding into something that other people can pick up from. And we sort of came together around, okay, what are the feasible things that we could do? We started to register the observatories. Again, this at the moment it's quite loose. It's mostly through our membership in Globicon, but we drafted a registry service, which we are talking to several other international data repositories that may be able to host that. We started to create more formal links to UNESCO IOC to feed into the essential ocean variables and GeoBond for the essential biodiversity variables. Some meetings followed from that. We started to exchange standards, not just information standards, but also actual physical samples between the nodes so that there can be some calibration between what must be, we have local constraints, so methodologies will be different for the moment, and we just want to see how that system reacts. Digitization and sharing of protocols, of course, and coordination of our bioinformatics code through GitHub, just in the Glomicon organization, nothing heavy, nothing we can't maintain, basically just keeping track of things and orienting people towards a guiding kind of principle. And then working with the GSC, thinking about how we can update existing standards to match this now more operational reality. I won't say much more about that, but pick up on that a little bit later. So it was scoped, so this is timeline so far. It was scoped in the end of 2017. Glomicon was set up through 2018 with partners from Atlantis and Agriculture and Agri-Food Canada. We started to align with some international frameworks, in this case UNESCO GOOSE, creating an essential ocean variable for microbes and propagating omics with the other panel members who are of course already tuned into that, and thinking about how we can do that. And then in partnership with Future Earth and NCEAS, we started to think about how to carry that forward for a global observation system from whales to microbes using omics and eDNA technology as part of a combined strategy. What we're up to now is thinking about how we can take Glomicon into the future, merging it with existing, let's say, frameworks that connect academics and operations to policy, like GeoBond, MBON, and others, and creating an omic bond as a thematic consulting network across all of the other biodiversity observation networks. So that's what we're thinking about now, putting together that proposal and pulling together more interest. So that's, I think, a very important message, because when we started to do that, then together with Neil Davies, Chris Meyer, and others, we had meetings where we're thinking about, okay, let's merge these activities that have popped up from various projects. We can't be territorial about this because we're building something bigger. So whenever we have the opportunity to merge and create something more ambitious, let's do that. Let's redefine priorities and really connect to a global system. It sounds ambitious, but frankly, that's what's being asked for. So it's worth tackling it on. So in the end of the day, it'll look something like this. We'll have a network of omic or microbial observatories around the world, coordinating their best practices in some manner, and also lots of legacy data, which will need a whole army of data archaeologists to trawl through. But the new data streams, at least, can come in and be distributed across the observatories through brokerage services and organizations using fair data technologies. I'm sure many of you are familiar with that. But again, things have to be interoperable. It's not enough to simply release things. Other people must be able to act on them and know what's inside them. And machines ultimately must be able to do that independently, because this is going to get bigger and bigger. Once that's done, we need to package that data so it can be shipped around in this space. And to some degree, that's already done, but there's much more work to do. And once we have that, then we can trigger machine agents to start looking across that, doing analyses that we could never even think of, and informing humans about what's going on out there in a set of virtual observatories that can be constructed on the fly almost. So that's part of the vision. And once we have that, we can start exporting collectively global data products into things like the Living Planet Index, GOOSE, GeoBond, etc. People who are asking for these combined data products. Okay, it seems like, okay, this is a lot of things we could do, but I'm encouraged because many people are doing this. They're doing this, but they're not doing this at scale. And scale can only be realized, in this case, I think, through the coordination of resources and alignment of vision between different regions, nations, institutes, even individual groups. So there's a lot of work to do there. Once we generate that, the knowledge that comes back into that will go back to the entire observatory community and say, okay, what do we need to change? Where needs more sampling, deeper sequencing? Which indicators do we need to develop? For what purposes? And we of course have to engage broader communities, both within omics and beyond. So that's the sort of architecture that we're thinking of and thinking of taking forward. So what are we doing in terms of engaging the community? Jerry already mentioned a little bit of that. I'll pick it up. There have been a number of meetings. I'll only highlight 2 here. The Genomic Standards Consortium, their 21st meeting, we had a satellite meeting where we brought together what should be competitors, really, in Vienna, and said, okay, this doesn't make sense. Of course, competition will always occur, but let's compete within a collaborative framework and build something. And that was where we decided, all right, let's merge GOMICON and the Genomic Observatories community in a new model so that all stakeholders, their needs are met and we have to redraft. And that's been a very, very exciting process. It took some time to get some money together to further that on, but now some resources are emerging through some European and also US projects, and we're activating that throughout this year. As Jerry mentioned, we pulled together some actors at Biodiversity Next. So ELIXIR was there, GLOMICON representatives, Agriculture and Agri-Foods Canada, among others, and had a session that talked very much about this theme, but also in some of the data sessions, we talked about interoperability solutions. So really biodiversity standards to enable the broader scale interoperation. So that was very promising, and there's been some good follow-up work from that. It's not just a question of outreach. We also have to connect action. We have to connect this to action to show that, look, this could work. This is tricky because this is not often directly funded as a collective global action. It's very hard to bridge so many stakeholders in one go. So a lot of this is on the goodwill of the participants and the interest in saying, okay, let's try to realize this together. And I find that encouraging because that leads to funding, and indeed it has already led to good grant proposals that have gone in, and some of which have been successful. One of the key activities is linking up 6 observatories. Well, we started with 6. I believe maybe 1 or 2 more have joined in, but actually exchanging physical samples between them, as well as mock community DNA extracts. So essentially synthetic communities. We know the composition. That's our baseline. Again, back in Atlantos, that's what our work package leader, an oceanographer, said. You need to send a known quantity to people so that we can calibrate properly. And this was exciting. So we did a sort of round-robin exchange, including the mock standards sent out from the AVI to the other participants. And we're getting results back now. Logistics of this were surprisingly difficult, but Kathleen and Felix did an incredible job of coordinating all of that and making it come together. And so that's happening now. This is going to be very interesting because each one of these observatories have very different methods of handling the samples, sequencing, processing, informatics. And we just want to see, do we get any robust signals out of this? We need that sort of reality check. And I like this because this is different from saying, okay, let's standardize it this time to publish a paper. Okay, that's really interesting. That's very cool. But I'd rather see what's actually happening day to day and what do we need to do to make this then a sustainably interoperating system. So that's the spirit there. On the data side, again, picking up from ELIXIR activity, so we had a workshop with ELIXIR, Silva, Magnify, the ENA, so the European INSDC Hub, and GBIF, with their work, primarily GBIF, ENA, and ELIXIR, and with Magnify's compute ability and Silva's databases, to pull in all of the sequence information that's been georeferenced, that has taxonomic assignments possible, and create a global map. And this is the thing that a lot of stakeholders want to see. They're not really going to be able to read your papers. They're not really going to be able to get into the data itself, but they want a kind of overview of what's going on. A policymaker would very likely want to see this, and then products distilled from this to say, where are the invasive species? Where are the potential pathogens? Where are the, and even if it's not perfect, there are many things here that, our methods are not standardized, etc. But that's true for other biodiversity communities too. But you can still make decisions that could potentially save resources, lives, you name it, right? So even if we get 60 to 70% of the way there, I think that's something to get out there. And this partnership is one mechanism to do that. Some work that's happening around here, just highlighting one of my master's students who is brave enough brave enough to take on this space. has been working with a collection of observatories. In this case, it's from AVI, the Hawaii Ocean Time Series, and the San Pedro Ocean Time Series. We did have data coming in and a lot of support from others like IMOS in Australia. In the time frame, however, that she has available, we had to prioritize. And essentially, we're leveraging some of their data products in the long term, using the GSE standards like the biome format to package the metadata, taxonomy, and tabular data, and simulating what an operational system of observatories sharing data products would look like. So we're trying that, and we also talk to the developers of the biome format. These are not dead standards, they're very much living. In consultation, in this case with Obis, Ward and Peter, and Daniel McDonald, the lead developer of biome, to say, how can we update that to make it more fit for purpose in an observatory setting? You know, we need to know what code you used. We need data integrity checks to make sure that everything's good. We need to know your software version so that you can reproduce things exactly, especially if you need to recreate a data set. There are going to be legal repercussions in some cases. So that was discussed, and they're on board, and new releases are being made, and we hope to continue that partnership. And once you have that, once these things start talking to each other, you can start creating dashboards of alpha diversity, beta diversity, looking at seasonal change across the partners. This was a small simulation, but imagine this at scale. Imagine this networking all of the 100 or so observatories around the world and many more coming in, allowing us to automatically discover signals that require human attention or that are interesting or coincidences or errors. So there's a lot of potential there. And that's what Raisa will be generating for her thesis, which will be submitted later this week. Good fun. We're also talking to developers of data science journals. So PennSoft has been supporting the biodiversity information standard space for a long time. And we had a meeting with them to create a journal to allow people to publish artifacts about the data that they submit in an interoperable way. Why do we need this? Well, there needs to be an incentive for the scientific community to release their data early, as opposed to sit on it until they can write a high impact paper. It's a reality, you know, this is how until funding agencies change their, let's say, incentive structure, which is a discussion that's happening. We need to incentivize the science community, who are the sort of custodians of a lot of this data, to get it out there as early as possible, and also they need credit for it. So that's another aspect of it. So with that, we also have a set of increasingly aligned projects. I'll give a couple of examples here, again with a European bias. So all of this stuff that I was talking about, it started as a kind of wouldn't it be great sort of mode. But we wrote projects based on this. And for example, one that came through, EuroSea, which involves GOOSE, so the Global Ocean Observing System, that's funded. It's a medium-sized H2020 project. And it's really pulling together European marine observers in a way to create an interdisciplinary ocean observing system, including biology. Biology is always underrepresented in the oceanography realm, but that's largely because we need to operationalize. It's really hard to work with something that's kind of squishy, as biology tends to do. That also applies to the data that comes out of it. So we're trying to formalize that. We're trying with the things that you've seen and many others to pull it in, and also omics and eDNA are a large part of that. In particular, us at AVI and also our collaborators, Daniele Iuriccioni at SCN, Stazione Zoologica Anton Dorn, we're going to continue the GOMACON mission and evolve that with the Genomic Observatories network, merging it into the omics spawn, and also link that to testing standardized protocols that are partly the legacy of the Tara Oceans expedition, because again, they needed to operationalize for that large-scale expedition. And our job at AVI is to link that to the activities in the GOOSE BioEco panel. So I lead the microbial essential ocean variable there and discuss with the other panelists how to integrate data, and especially omics data, across different kinds of living systems. So that's our goal there. Another project that came through recently is run by Obis, so Ward and Peter out of Obis, which is looking at a bioinvasions observation system. So again, using eDNA omics technologies to detect bioinvasions and protect genetic heritage within ecosystems and monitor their stability. It's really key to have stakeholders like Obis involved, because the discussions that they have are focused more, let's say, towards the UNESCO IOC realm. They're thinking about the Sustainable Development Goals. They're thinking not just about publishing papers, but delivering products in that space. So, for example, the work packages that are dealing with decision support systems, that are reaching out to different stakeholders in policy, the public, etc. That's a kind of, let's say, socio-technical expertise that we need to bring much more into the realm of, let's say, the pure innovation and research. So that's really quite a remarkable thing to be working on in the next year or so, and also pulling in all of the other resources we just talked about, networks, resources, and opportunities. This is a slide from Ward Appleton's explaining how that all happened. So again, it's really focusing on the data and the infrastructure, bioinformatics, quality sampling. It's not light on that at all, but it flows into decision support systems for other stakeholders. It flows into existing habitat suitability models, and that's that mainstreaming aspect which we really want to culture it for the future. Right. Lastly, we have to talk about the global frameworks. So we have projects going, we have local activities, we have demonstrators and prototypes, but there are global frameworks afoot that are becoming more aware of this, and those are conversations that need to be had such that, again, we have to do this in a fairly diplomatic way. It's not promoting one project or another project. It's a sort of rising tide that must lift all ships, and largely that, for my international work, that's been my position here. We have to kind of make this this competition within a collaborative framework a reality. So I'll highlight a few things. Of course, we've mentioned GOOSE, the Global Ocean Observing System, currently run out of UNESCO, and they're linking science observation in terms of operations and the development of projects to support that goal. They have a series of regional alliances that span most of the world's oceans. And in order to start to streamline the data flows and also the topics of inquiry for the observing system, they've been running the essential ocean variables for a number of years now. The EOVs are an opportunity to link monitoring with a set of best practices that are focused, that are targeted, that are reproducible and implementable in different settings, and also that have been written in a way that they can be taken up by lower capacity nations. I mean, if we want a global system, we cannot chase the cusp of innovation all the time. We do also need to scale it back and make sure others can participate. So those EOVs nest within another whole set of essential variables to keep tabs on the entire planet. Climate variables, weather from WMO, the essential biodiversity variables taking shape under GEO. And there's an overlap between them all. And that's where the biology stuff happens. So where the essential biodiversity variables and the essential ocean variables overlap, there's a lot to do in terms of coordinating these global frameworks. And there have been this whole series of meetings that I won't go into, but indeed, gradually, it's a nudge theory sort of thing. Things are coming together. People are aware of each other. It's just a question of how do we act on it. And I'm hoping that a lot of the projects and implementation work that we're doing now can matter to do that. So MBON is a device. So the Marine Biological Observation Network is again a device and network that's catalyzing that link between the EBVs and EOVs, and a place where partnerships can be built and furthered. We're looking to MBON for guidance, and they've been providing guidance on how to draft this omics bond proposal and build that sort of global federation. So I mean, I bring in Star Trek because I feel like I have to. But ultimately, that's where we have to go with the essential ocean variables. Like if you're on your spaceship somewhere and you find a new planet, what's the captain in that ship going to see in terms of a briefing about whether it's suitable? Where are the opportunities, the resources, the risks, etc. And that's really how I'm thinking about. So I'm a data scientist at the end of the day, and so that's how I kind of think about what we're building. This whole architecture that we're constructing around this must deliver streamlined and usable data products that are tailored to the recipient. So it's going to be quite a system. So there it is. The microbial EOV, which then I coordinate, is nested with a whole set of others, and others which have much more mature observing networks already existing. And then how do we connect that and mainstream not only different compartments of life, but also different ways of observing that life, including omics. I mean, it's quite easy for microbes because whenever we look at any kind of diversity aspect of microbes, you're going to get some kind of omics in there. And this is true for other compartments too. It's just a question of now socializing that across the networks of networks. Another thing that came up very recently, we were at the All Atlantic Forum at the European Commission a few weeks ago, and the Atlantic Ocean Research Alliance, or AORA, it's a European cost action, has been supporting the creation of a European marine microbiome roadmap that we delivered to the European Commission during this meeting. These are just some slides that we communicated to the Commission and others there. And it's the same sort of thing. And that's really encouraging because some of the text inside that roadmap that was delivered has been text we've been developing since 2016, 2017 at meetings about augmented omics observatories during the G7 process. So it's encouraging to see that it's materialized, it's taken shape. Others have recognized its importance and helped us shape material that can be understood by other stakeholders. That's us handing over the roadmap over there. So the initiative was led by the Icelandic contingent of ERA and coordinated by Margaret Ray from the Marine Institute in Ireland. So quite a lot of the usual suspects there and some new ones. That group is also active in the discussions of the upcoming UN Decade of Ocean Science for Sustainable Development, because funders are tuned into that, scientists are, policymakers, many are. So that's us working late at night in Halifax, in this case, the North Atlantic Regional Alliance, a meeting of the UN Decade, to develop the marine microbiome roadmap, and that includes omics technology and eDNA technologies and approaches, which are on the radar. They're very much on the radar. You will have fairly senior, high-level individuals calling for this, but what they're calling for is something that we have to shape. There's a need, and that need could either be filled by a sort of gold rush of, the biggest project takes it, or we can try something different and try to build a global interoperating system. I'm a fan of the latter, in case you haven't guessed. Right, so that's it. I'm just going to wrap up there because, this is just the tip of the iceberg. There's so much more activity going on, not just in, let's say, the Atlantic basin where you have more developed countries, but in many others. And what we need to think about is how to make it more normal for global sustained solutions to be built. And that means every project can have its lifespan. Everything doesn't have to become an infrastructure, but the outputs have to be absorbed by some kind of infrastructure so we have sustained solutions generating sustained and predictable data products that can be changed over time, but again, through a process that allows us to intercalibrate between data series so we don't break time series, and that we can calibrate between them. This is not new by any means. This is logic that's been in oceanography and other fields for a long time, but because of how innovative the omics and eDNA space was, sometimes we forget that. So having operational partners there is really key. So I just stress that, we have all the ingredients we need. We just need that vision, that socialization, that kind of common implementation strategy, and ability to work with each other across projects. And I'm more than happy to catalyze that and make introductions if necessary to bring this community together and build something that we can really contribute to the Earth observing system at the end of the day. One thing that came out actually when drafting that paper, the marine microbes paper, we talked about monitoring a lot. And we were told by one of the reviewers that, monitoring is a kind of dirty word in science almost. We don't monitor, we innovate. But then, as time went by, I thought, well, if we want to address some of the largest questions in science, we need long-term data series. Monitoring is part of science, and it's something that we've been communicating to also funding bodies and others that we've been talking to, that if we do want to tackle the challenges put forth in the UN Decade, for example, or other things, even national implementation strategies, regional, like the European Marine Strategy Framework Directive and others, we do need that baseline. We do need that trusted foundation of knowledge and data where we can start asking much bigger scientific questions. So with that, I'd like to end. There are far too many people to name. I put a bunch of logos here. I could put much more, but my brain is like 40% acronyms at this stage. I'm very sorry if your logo is not there. Just for the audience, just know that there are lots of people to thank, and everyone, I think, detects there's something here, and it's on us to make it a reality. So thank you, and we have 20 minutes for questions, I believe.
Thanks to Pier Luigi. I thought that was an excellent talk and really, really useful, very informative. So I've got a question. You talked a little bit about how this is now on a point of scaling up, and you kind of explained how that's going to happen within all of the observatories and so on, and the level of data that's being collected. What do you see the impact on systems like GBIF, which you also talked about, in terms of the amount of data that's going to be exposed in GBIF that arises from molecular type data? If you look today, there's about 20 million records in GBIF that have arisen from sequence data. How do you see that scaling over the next year, 5 years, that sort of time scale?
Mm-hmm. Yeah, I think that's a really important question because, again, there's this huge enthusiasm, and omics can generate mountains of data. Thankfully, we have the INSDC resources in place to handle the raw sequence data. And we have systems based at the EBI like Magnify that has the server farms capable of processing that into products that are then usable by stakeholders like GBIF and OBIS. They can pick up the taxonomy strings, they know that there's been some quality control, and they can package that for their users. I think what needs to be done there is a little bit of conceptualization work, because a record is not the same as necessarily observing an organism in the wild. We're talking about compositional data, we're talking about a different kind of life data. So I think there needs to be a little bit of conceptual work there into what is a record, what does get forward, how can we merge records in the sense, if we have multiple presence-absence detecting, how do we bundle that together to say that, okay, this is probably what's happening there, especially with things like eDNA, let's say of fish stocks or something of that kind, because we still need to calibrate on how long is that viable in the environment. Is it degraded? What kind of signal are we looking at? Is it the same in different ocean compartments? It's not, right? So it really depends on a lot of calibration. So I think a little bit of caution, but at the same time showing possibility is the right balance there. But in short, we still need to do some conceptual work before we get carried away. Because if we get it too wrong, I mean, it's okay to get it a little bit wrong. I mean, everything's growing, everyone's learning. But if we miss the mark and just shove data at people, it's very impressive. But as soon as it starts making predictions that are nonsense, then people are going to get turned off, right? So we do need to think about how to do some quality control. That's my stake on it.